The transcription denotes what exactly? The RNA synthesis process in the molecular base of inheritance. So why the RNA synthesis should supposed to happen? We know very well, if the RNA is not there, ribosome is not there. If the RNA is not there, ribosome is not there. If the RNA is not there, RNA maturation is not there. If RNA is not there, protein is not there. Okay, if protein is not there, you are not there, I am not there, right? So here, if I need a protein, I need the RNA. If I need the RNA, I need the DNA. Okay, here protein, RNA, and DNA, right? So if I need the protein, I need the RNA. If I need the RNA, I need the DNA. This is the way you are understanding, right? So basically, what is the enzyme involved in this? RNA polymerase enzyme. Okay, the enzyme which is involved is DNA dependent RNA polymerase or RNA polymerase. In multiple way, they'll bring the confusion. Be what? Very careful while saying the answer. They will confuse you in many ways. DNA dependent DNA polymerase, RNA dependent DNA polymerase, DNA dependent RNA polymerase. Many enzymes of different name, what I told to you, are having different type of function. Okay. Now we are entering into the next important structure, double-stranded DNA. If we're talking about the double-stranded DNA, of course, is becoming a single-stranded DNA by using that a single-strand RNA is synthesized, which is very important for making the protein of various quality and quantity, right? So we know very well the double-stranded RNA we are finding, for example, in the case of rotavirus. In the case of rotavirus, of course, we are finding double-strand RNA. This double-strand RNA formation is not the case in the case of human being. In human being, double-strand RNA is not there. In HIV, there is no double-strand RNA. Okay, only in few virus like rotavirus is having double-strand RNA. That means the unwinded DNA, both are not participating in the RNA synthesis. Only one single-strand DNA. Only one single-strand DNA is making the RNA. That is a message we need to understand. Now we are coming for the next phase is transcription unit, which is nothing but a unit which is responsible for making the RNA synthesis to happen. So what I'm saying for you is, I'll give the better clarity for you. Can you see this? So in this picture, if you're observing the template DNA, structural DNA, and then coding DNA strand. So we know very well, always this is a transcription unit. This is a transcription unit. So template strand is nothing but a parental strand. By using this template strand or parental strand, new DNA strand is synthesized. So we know very well the template strand is having the coding sequence, whereas the coding strand is not having the coding sequence. Right? So always the RNA synthesis happens the same way how the DNA synthesis happens. Okay. Here 3' to 5', 3' to 5'. Okay, DNA template is 1 with polarity of 3' to 5'. Right. So what we need to understand basically is the RNA sequence is this. This is RNA sequence without thymine, right? So the template strand having coding sequence makes the new RNA. Template strand with the new coding sequence makes a new RNA. So this is the way you are understanding the message. Now we are entering into the most important concept, which is nothing but the transcription process. When we are examining it, okay, 5' end is upstream point and 3' is downstream. Okay, downstream. Can you see this? The new RNA is getting synthesized. By using the DNA information. In this picture, can you see this? The unwinded DNA, both are not participating. Only one DNA is participating in the RNA synthesis. Have the adamant clarity. Have the strict clarity. Okay. The other strand also may, and all don't have this type of diplomatic mind, and be very strict in conveying the message. Okay, now we are coming for the next phase explanation is um, the non-template DNA, which is not a which is not a which is not a DNA for making, which is not the DNA for making RNA. So this non-template strand, also called as coding strand, whereas the template strand is in the coding sequence. Template strand is in the coding sequence. So this non-template strand is a coding strand for whom? For parental strand. What do you mean by parental strand? Template strand. What is this template strand makes? New RNA. What is template strand makes? New RNA. Are you getting my point or not? So now we are coming for the next phase explanation is here. Before entering into the next subtopic, okay, the questions we'll discuss. This is one of the classic questions. Okay, and this question we already discussed. The option is C is the correct answer, right? So now we are entering into the other question. How many types of RNA polymerase are there in the eukaryotic cell? Of course, three. Three type of RNA polymerase are there. So the first and second question, both are choice questions, which are repeatedly asked n number of times for the past 10 years. This is the way we need to narrate the answer, right? I already told three RNA polymerase, 1, 2, and 3, right? So RNA polymerase 2, responsible for bringing the maturity in the RNA, right? So like this, what we are bringing all the RNA polymerase information in detail, right? So now we are coming for the next messages. Here, if you're talking about the next important question in the which of the following RNA carries amino acid to the ribosome during protein synthesis? Of course, mRNA. No, tRNA is a correct answer. tRNA is a correct answer. Can I see this please here? Okay, tRNA is a correct answer. Right. So now we are coming for the next phase explanation is okay, what is the work of the RNA polymerase during transcription? We know very well the RNA polymerase catalyzes the process of transcription by synthesizing RNA strand by using DNA template. By using DNA template. This is the correct answer. So now we are coming for the fifth question. The fifth question. RNA polymerase reads the DNA template during transcription. Okay. In 3' to 5' direction. 3' to 5' direction. And synthesizes new RNA. And synthesizes new RNA. This is the way you are remembering, right? So now see again back I'm repeating. So always always you need to understand. Okay, either you take RNA synthesis or either you take DNA synthesis. Always it happens from 5' to 3'N, right? So this year they asked a DNA template. Next year they will ask RNA template. So be very careful. That means in the upcoming board exam, related videos, whenever you're having time, look into this in our GTU portal, Gotouniversity portal, tips for exam, understanding all the basics and terminology and enzymes and many sub-steps with the diagram along the labeling is very important. That is the tips for the exam. How promoter works, how the terminator works, how the enzyme works, okay, how the sigma factor works. Okay, like this if you're asking question, so you can bring all the concepts very carefully. Now we are coming for the presentation, transcription unit. This transcription unit, here, be very careful with the transcription unit. We know very well the transcription unit in the sense some RNA can make single protein, some RNA can make many proteins. If that is the case, I am an RNA, I will make single protein, that means monocystonic. If I'm making many protein, polycystonic, right? Now we are coming for split genes in the eukaryote. What do you mean about split genes? Split genes, basically, it is very, very important in the eukaryote. Okay, split genes. Okay, structural genes are often called as split genes. The coding sequence are interrupted by the non-coding sequence. Non-coding sequence. I am a DNA. I can't do anything. I am mum. I am a dummy DNA. That is the way the non-coding sequence are communicating, right? Okay, non-coding sequence. Now we are coming for the next two phases. The non-coding sequence we used to call as intron. I can't do anything. Okay, just as a mom, I'll sit. I give only the instruction to various people. You do this, but I won't do any work. Like this, certain introns are existing like this. Okay, that introns which has to be removed to make the mature RNA, right? So this is the way you're remembering. And now we're going for the types of RNA. Before going to the types of RNA, anybody having any doubt, please let me know. I'm there with you to help you. We know different type of RNAs. One is called as mRNA, another is called tRNA, another is called rRNA, right? So you know very well the RNA polymerase makes different type of RNAs. One is called as tRNA, mRNA, or RNA, right? This RNA polymerase is behind the sigma factor, which makes the RNA polymerase to get attached to the DNA so that the RNA polymerase can do the new RNA synthesis. This is the crux of this particular multiple sentence, or conclusion of this multiple sentence, or the essence or the core of this multiple sentence. Now, this introns, when it is getting removed, what happens exactly? The mature RNA is formed. Who is playing this role? The RNA polymerase plays this role. As a result, you are getting mature RNA. Now we are coming for the transport of the mRNA. mRNA is getting movement into the ribosome for making the protein, right?